Hello ma'am, my name is Mohammad Yunus Khan and I am here to present this paper with my group members. So the paper is about short-term metabolome dynamics and carbon, electron and ATP balances in chemo state grown Saccharomyces cerevisiae. So here is my introduction part. Intro of this paper is defining us the importance of metabolic model which is used to understand the enzyme kinetics in microorganism. So the metabolic control mechanism is operated by the level of metabolome. So here I want to tell you little about what is metabolome. The metabolome refer to the complete set of small molecule chemical found with the sample like cell organelles or tissues. It can also be used to assist the rational designing of metabolic pathways to enhance desired functionalities of microbes. In this paper, we also see the kinetic parameters in this kind of model can be obtained by stimulus response, in which cell grow in quasi-steady state, are perturbed by an external stimulus or dynamic response of intra and extracellular metabolite, which are monitored in this process. So, in this paper, we are also seeing the importance of various metabolic processes in kinetic model can be evaluated by the relative contribution of metabolites which is involved in these processes to the overall material balance which form the basis for kinetic parameter estimation. Under the quasi steady state condition, for example material balances of extracellular metabolites which are required for the metabolic flux analysis. So this information will increase our understanding of short-term physiological response of Saccharomyces cerevisiae to glucose excess and will assist in building more realistic and comprehensive kinetic model. So there is four objective of this study. The first objective is to quantify an extended range of metabolome responsible for glucose pulse to aerobic carbon limited chemostat culture of Saccharomyces cerevisiae. The another objective of the study is to construct complete carbon electron and energy balances during a 300-second post plus period from this measurement. The third objective of the study is to identify major carbon and energy production in the process. And the fourth objective is to determine if major unquantified metabolic processes are remain. From here, the further part is given by Priyanshee.
Hello ma'am, I'm Priyanshee and my part in this research paper is materials and methods. So there are several methods which are performed in this research paper sequentially. So first is yeast strain and chemostat cultivation. Second is rapid sampling, quenching and metabolite extraction. Third is determination of culture dry weight. Fourth is extracellular metabolite analysis. Fifth is mass spectrometric analysis of intracellular metabolites. Sixth is intracellular trihalose analysis, and seventh is the intracellular glycogen analysis. Some of the methods are explained by me and some of the methods are explained by my friend Aashu. So first is the yeast strain cultivation and its chemostasis. So the yeast strain is cultivated in anaerobic and carbon limited conditions. Then the cultivation is mixed with glucose and ethanol to obtain the biomass in steady state. Now the dissolved oxygen tension is analyzed along with the oxygen and carbon dioxide contents. The DOT is maintained above 70% in the steady state. The second method is rapid sampling, quenching and metabolite extractions. So the supernatant is cultured for the analysis of extracellular metabolites, and after that it's rapidly quenched. The filtration is performed afterwards. Analysis of intracellular metabolites is carried out. Broth is rapidly quenched with methanol and water. After centrifugation is performed, it is mixed with the pellet is mixed with methanol and water and centrifuged again. The intracellular metabolites were extracted from the pellet. The ethanol is then evaporated to dryness. The dried samples are deionized and resuspended in deionized water and centrifuged again. The supernatant and pellet were separated and stored at -30 degrees Celsius and then analyzed. The third method is determination of culture dry weight. The cultured samples are filtered through nitrocellulose filters which are pre-dyed and tiered. The filters are washed twice with demineralized water and dried at 70 degrees Celsius and then weigh carefully. The extracellular metabolite analysis is the fourth step. The amount of glucose, acetate, and glycerol in the supernatant is measured with the enzymatic kits. The concentration of ethanol was analyzed with enzymatic bioanalysis kit. The absorbance is read on UV spectrometry system. Now the next part is going to be performed by my friend Aashu.
Hello ma'am, my name is Aashu and I will be presenting the further points of materials and methods. So first point that I'm beginning with is MS analysis of intracellular metabolites. So the concentrations involved in the intermediate complexes of glycolysis cycle were analyzed by liquid chromatography, electrospray ionization, tandem mass spectroscopy, etc. The same analysis were applied on analysis of complexes involved in TCA cycle. The concentration of only adenine nucleotides were quantified and separated. Chromatography is performed at 20 degrees Celsius and the solutions are filtered to use the remove particles and bubbles using HPLC instrument. The next point is intracellular tree halos analysis, which says that intracellular tree halos are analyzed by quantitative nuclear magnetic resonance. Then lyophilization is performed, which is a process of cold drying. Then the content of tree halos is calculated. Next point is intracellular glycogen analysis. We found intracellular glycogen in the cell extracts and insoluble residues, which are hydrolyzed by amyloglucosidase without alkaline extraction. Thank you.
Hello, I am Puranjaya and I will be explaining results section. First, changes in metabolic fluxes and intracellular metabolite following a glucose pulse. This table shows the average variation in the accumulation of extracellular carbon sinks and group of intracellular carbon sinks relative to steady state level, which are within 10% and 15% respectively. The figure explains increased in the residual glucose concentration from 0.15 to 5.56 millimolar, lead to dynamic in the biomass specific glucose uptake rate. Glucose, the secretion of ethanol, acetate, and a small amount of glycerol, and dynamic changes in the biomass specific rates of O2 uptake and CO2 evolution. Notably, both QO2 and CO2 are closely related to flux through the TCA cycle, which is a major source of NADH and non-fermentative CO2 and accounts for 69% and 82% of steady state QO2 and QCO2 respectively. They divided the 300-second post-pulse period into three sequential metabolic phases that are characterized by distinct metabolic responses for a number of metabolites. In phase 1, highly dynamic changes occurred in metabolic fluxes and intracellular metabolite concentrations. In phase 2, gradual changes in the extracellular fluxes and intracellular metabolite concentration were observed. In phase 3, the extracellular fluxes and metabolite concentration remained rather constant. Second, cumulative carbon, electron, and ATP balances. In phase 1, intense dynamic in glucose glycolysis lead to elevated pool of phosphorylated compound and storage material, leading to a large energy drain and possibility decrease in Q anabolism. In phase 2, a gap between the carbon electron source sinks and emerges at about 100 seconds. In phase 3, the difference between the carbon electron source and sinks continue to increase. Third, intracellular free and bound phosphate. Increased concentration of phosphorylated intermediate are compensated for by decreased concentration of adenine nucleotide and PI, leading to a relatively constant amount of the measured free and bound phosphate in the cell during the post-pulse period. This implies that the hydrolysis of pyro and polyphosphate is not significant during the post-pulse period of energy generation.
So, it can be concluded from this study that the metabolic responses observed here were different from those observed in the similar glucose pulse experiment. And the possible reasons for this difference could be the strain of S. cerevisiae used, the growth rate, and the carbon source. The difference in storage carbon metabolism could be due to mutation in adenylate cyclase in the yeast. In addition to enzyme metabolite interaction, protein phosphorylation needs to be taken into account for the observed increase in fructose 2,6-bisphosphate. Also, in phase 1, glucose transport is subject to significant inhibition. In the phase 2, the concentration of glucose 6-phosphate, fructose 6-phosphate, and fructose 1,6-bisphosphate decreased while Q-glucose increased. So, it implies that the stimulatory effects leading to the increased fluxes could be a recovered energy charge in the phase 2. The increased anabolism implies the excess of ribosomal proteins and low ribosomal efficiency. So, concludingly, this experimental study provides a comprehensive picture of the extra and intracellular dynamic response of S-ceribicide, and it emphasizes the importance of including storage and anabolic processes in kinetic models.